Okay. Yeah, so today I'm going to talk about hashed embedding. So it's an embedding technique for protein sequences. So the overall idea is that we sort of find some numerical feature representation for protein sequences, and then we can use machine learning algorithms downstream to do all sorts of tasks, right? Like clustering, classification, whatever we want, right? And this is a work with my students Taslim Murad and Sarvan Ali, who couldn't join us. So I'm going to give a talk in their stead, and so this is what I'll talk about. And so the motivation is sort of protein sequence analysis is becoming it's always important for understanding different biological processes and how to understand the diversity and dynamics of organisms or especially viruses, I guess, especially with this SARS-CoV-2 outbreak. I mean, we've sort of done SARS-CoV-2 to death, I suppose, in some sense. So we do this analysis on SARS-CoV-2, but it is a general purpose approach. So it's really an approach for embedding sequences into some numerical feature representation to allow machine learning algorithms to analyze it. So it could really be used for anything, but we do it for SARS-CoV-2 because it's a large data source that we can study and see how it performs, right? So and so it is sort of interesting with all the sequence. Yeah, like the SARS-CoV-2 is sort of interesting because it I think right now on databases like you say, there's tens of millions of sequences available, right? So it really renders a lot of tools obsolete for its analysis, or maybe not obsolete, but it really provides a bottleneck for such tools, and so we try to go at it with a machine learning sort of approach to study the virus, right? So and so machine learning models are good for this, for this sort of big data application, let's say. However, they require the inputs to be in numerical form. So there's a lot of sort of need for techniques to convert these into this numerical form for downstream analysis with machine learning approaches, right? So so we will study the SARS-CoV-2 virus, and in that sense, what we're going to do is just focus on the spike protein. It's sort of a biologically meaningful dimensionality reduction technique, you could say, right? Instead of focusing on the entire sequence, we just focus on this spike region because maybe I don't know how true this is anymore, but a lot of mutations are happening disproportionately in this spike region, and it's also those mutations that are important for mediating interface with the host cell membrane. So it's like a lot of these variants for that we're able to evade immune response and stuff like that. It's all mutations happening on this spike region, right? So we sort of focused in on that. Maybe another reason too is that the GISA database provides like the spike sequences directly, so it's kind of it's convenient for that reason as well. So what we do is and I'll go into details of the proposed approach, but what we propose a hashing-based method to generate a fixed length alignment-free embedding of a protein sequence, and that's a lot to unpack. So I'll unpack each of these as we go. What it does is, so what we do first is we generate k-mers of the protein sequence. that's how it's alignment free, because we're just generating the k-mers, and then and then we're computing weights for these k-mers, and then kind of building a weighted frequency vector of it based on a hash function. I'll go into all of this in more detail as we go. So, any questions so far? I mean, just feel free to interrupt if you have any, or as you prefer. And sort of the baselines that we're going to compare to, we compare to a list of the following baseline. A lot of these of our own are quite a few of these are, well, I guess just ICCABS and PWM are our own methods. So we've been kind of playing around with a lot of these different embedding approaches to computing fixed length representations of feature vector representations of molecular sequences, I guess you could say in general, right? So SpikeDevec is kind of a more basic one that's based on k-mers. PWM is also based on k-mers, but we use a position weight matrix to sort of assign weights to the with the k-mers, you're just really looking at the amino acids as an alphabet. The PWM is a position weight matrix that assigns different weights to different amino acids. So it gives you kind of a different a more nuanced sort of embedding. String kernel is another outside method. I don't know so much about that. I think it's just applied to strings, any sorts of strings. So what's interesting is these embedding methods can be used for all sorts of sequences, right? Like you could study texts with it, like comparing different versions of old texts. I mean, you could use it for music sequences, like you could. I think it has been done for you can take some music sequences and do some classification, and you'd be able to sort of cluster hip hop and country music and rock music and different sort of things, right? Because they have features that are kind of distinguish those, right? And then we use this Wasserstein distance-guided reinforcement learning, neural network-based approach. Space Kamer is sort of you're running space Kamers. You just run a template of consecutive, a window that kind of slides over and gets you consecutive amino acids. Space Kamer, you just kind of run a template that like first position, third position, and 10th position. There's, whatever sort of template you want, you run that over and generate Kamers. The auto encoder is another approach, and then ICCABS I don't know too much about, but sort of like there's drawbacks with all of these. There's the following drawbacks with all of these approaches, and and so to get into the proposed approach, so indeed first we compute Kamers. And so that's just with a sliding window over the sequence to generate a set of Kamers. And then we use the Blossom matrix to assign a score to those Kamers. To be honest, I don't fully understand the point of this, but it's sort of to assign a weight to you take a Kamer and then you run a window of size 2 over that Kamer and produce Blossom scores for neighboring amino acids in this Kamer. And that will, then you sum up those scores, and this gives you a weight for that Kamer. So it can represent some sort of we had another hashing method before, and we were just assigning a weight of 1 to the Kamers, and we thought we wanted to kind of vary this a little bit to assign some sort of importance to the Kamers, that's a little bit, yeah, we still have to suss out a little bit about what that means, but this is what we do at the moment. So we've computed the k-mers, we've assigned a weight to those k-mers, and now we're so the reason we sort of did this was, like, before it was something like spike to vec. You would just compute the k-mers, and then you would just get a frequency vector, which is just the k-mer counts, the counts of those k-mers. So in that sense, each k-mer has weight 1 in that sense, right? And you have a frequency vector, and that's your numerical representation, right? So it's simply that, right? But now we do that, and then we get the weight, and now we have this frequency vector, and then another, like, so with this frequency vector, another issue that comes up is sort of searching for the bin, right? Like, you have some k-mer, you have to put it in its proper bin, right? So, like, for example, if we had a 3-mer and it was AAT, we have to put, okay, if we had a 3-mer and we were studying DNA sequences and we had AAT, we'd have to, like, go and search for that bin, right? So like this array indexing can be sort of expensive from a computational standpoint, right? So what we do instead is we just use the power of hashing, so to give us sort of a constant time sort of access, right? And so what we, so we basically, like, we have the k-mer, and we use this SHA-256 hash to assign the k-mer to a bin, right? And in this sense, like, I guess, so the bins are, like, before the bins would be counts, but now it's kind of it's a weighted count in some sense. So we're assigning the k-mer to the weight of that k-mer to the bin, right? Using this SHA-256 hash function, right? And what's nice is that what's nice about the SHA-256 hash function is that it's deterministic. So the same input will always produce the same output. So reproducibility is there. So if we run it again, we get the same result. So it's fairly resistant to collisions. Like with these hashing approaches, we don't really resolve the collisions, actually, because at the end we're just producing a vector, right? So the downstream machine learning approach has to deal with that, right? I mean, it's not the end of the world if two things collide. You have a slightly suboptimal numerical representation that you're trying to perform this hash or clustering or classification upon, right? So I'm just going to check my time. I think I have it okay. And it's also, it's also a fairly, like, fast and sort of compact hashing. So it's you wouldn't want two sparse of a hashing, because you'd have a kind of a curse of dimensionality there, and it might not, like, machine learning algorithms can only perform so well, like, when it's really big. So I mean, we try to there's sort of I guess there's a trade-off between the number of collisions that we want, and sort of the dimensionality of the representation that we're producing, right? Or how big this hash table is. So that's sort of the proposed approach. And what we do it on is this SARS-CoV-2 protein sequencing data set. So I think this is a snapshot of the ESA database from, like, 2021. I'm not completely sure. Like, the alpha variant is sort of dominant there. So we're just going to use this as a proof of concept at this point to see if it's going to work out. One reason, I guess, is to compare to some of the other methods that were developed. We ran some experiments and saw how the performance of those were. So these experiments are just spoiler alert, these experiments are still running, so we're kind of, whatever comes out, we'll if they're good, we'll go for the post proceedings, and if not, yeah. So the models we are going to kind of compare to are as follows: support vector machine, naive Bayes, and so forth. And then we'll kind of just do a standard. So it will be classification that we're going to use to kind of assess the quality of these. So these evaluation metrics are in terms of the classification of these, right? So like accuracy. So I mean so basically we have sequences with labels. This approach would assign this approach would try to learn the labels from a training set, from like a training set, and then we would have a test set, and it would assign labels based on the sequences, and then of course it's going to assign the right label or the wrong label, and you can see you can get all your false positive, false negative, like those four values, right? False positive, false negative, true positive, true negative. And then accuracy, precision, recall, etc., are all based upon that, right? But yeah, the experiments are still running, so we cannot report the results. What I did want to say is we do have a previous, just so I don't have anything. We do have a previous paper that we did submit recently, that was accepted recently. On hashing Tvec, and it's has some similar ingredients in there. I think we use a different hashing function, and we don't do this weighting scheme. But and this one, so we tried this technique before. Even the pipeline can be sort of illustrated, I guess, like we're computing some k-mers and then assigning them here the weight is just 1, so we're just like binning them, and then we have this this hash table is your feature vector basically, right? As is the case over here as well, right? And so we ran that, and before I go to the results, as we expected, one thing that was really interesting is that the runtime was much less than this other method that we had developed before, right? So this was a PWM Tvec, and as we see, when compared to the baselines that we're using in this study, Hash and Tvec also outperformed in terms of accuracy and precision. And I do want to see the data set that we use. I think we use the same data set. I guess we'll compare to this one too. But we may have used several data sets, including I guess I'm running out of time. So with that in mind, so we generated an alignment-free feature vector representation based on hashing and k-mers, and the experiments are still running. And here's references that are sort of relevant, and that's it. So thank you very much. Any questions?
So it's I nice job, by the way.
Oh, thanks.
So the size of the feature vector is kind of fixed, and what's the relationship of it with the KMR or the character sizes of the sequence investigation?
Oh yes, so it's a fixed length feature vector because, like, for example, if you're doing three-mers, like say you're doing three-mers of an alphabet of size 20, so it's the length of that feature vector is going to be 20 to the power of 3, right? So it's or it's the other way around, 3 to the power of 20. Like so it's Yeah, it's gonna be 20 to the power of 3, yeah. Because you can have, like, for example, let's consider DNA alphabet of size 4, right? So if you had a 3-mer with an alphabet of size 4, it's gonna be 4 to the power of 3, right? Because you'll have a bin for AAA, a bin for AAC, a bin for AAG, a bin for AAT.
I mean, in that case, there's no chance of collision, right?
Yeah, if you're just doing the straight up binning, there's no chance for a collision. But then if you're but the problem is the bin search can be sort of expensive, so we just do hashing instead. So we'll assign, like, the hashing will take AAA and map it to something in this hash table, right? And then maybe AAC could also be mapped to that same location in the hash table, right?
So the final space has space. What is the size of it compared to the, like, 14 or 21?
Oh yeah. The size, like, I guess the size of it would be at most that, right? Because like, if nothing collided, it would be the same amount of space used by the bidding, right? Because obviously the hash function would assign the same K-mer to the same location all the time, but it may also assign 2 different K-mers to the same location. So it would be an onto mapping, right? So it would be, yeah. So like, yeah. So like the space would be about the same, but it's the time that we're really saving. It's a hash function that just, like, yeah, it's just hashing it rather than doing this lookup operation. So yeah.
Yeah, quite interesting. Thanks.
Oh yeah. Thank you. Yeah. Any other?
Yeah, I mean, kind of want to clarify one thing and it's connected to this. I think Daniel was an alphabet is a good example. You said 22 to the 3 different 3-mers, but most of them would not appear in your sequence. So you don't want to follow those.
That's
sequences.
another point. That's another
the idea of
point.
using the hashing, and that's just kind of wanted to make sure I understand. So you're hashing each K-mer, not the vector of counts. Yes.
That's a good point. Yes, we're saving even more space actually, because like if AAA doesn't even appear in your sequence, there's no But if in the binning situation, there'd be a bin dedicated to this possibility, right? But so you have this kind of very sparse vector anyway. And we do notice, yeah. So that's a good point. Yeah, that I.
The embedding itself, of different sequences. I mean, for example, you want to have locality of hashing, similar sequences should hash similar vectors. That's why the embeddings are useful.
Yeah.
But you, basically get that from the scenario that you have between the vector counts already.
Yeah.
Just look up those vectors.
That's a good point. Yeah, to be honest, really, like it's been mostly testing human server working on this, and they would attest to these sort of qualities. But yeah. Thank you.
Any other questions? Are you the next one?
Oh.